Okay, welcome everybody. Good afternoon, and welcome everybody for our weekly seminar series of the Department of Marine Geosciences. And I'm very honored to host one of our own people that grew up into a very prominent scientist, um, and Dr. Bialik from IOLR in Haifa. Bialik started his career, as much as I remember by heart. Dr. Bialik started his career at Ben Gurion University. Then he continued into Weizmann for a postdoc, then Princeton for a postdoc. Then he came to Haifa um, for a long period, and then he moved into cooperation with many scientists and based in both in Haifa and Malta and Germany. Um, did I forget something?
No, you're
Or
good.
I am okay?
Yeah.
And eventually he came back into to Israel for a position in IOLR. So we are very happy, or that you are back and that you are at home and that you are um, dealing with a lot of mud, which is great. So today, Or is going to, and aside of this career, Or is head editor of the depositional record, right? Of the
No, I'm not,
international.
no. I was just an interim editor-in-chief until they found a permanent one. I'm
Okay.
still an editor there though.
My point is that is filling up a lot of other positions, helping the academic community worldwide. Is involved in SED Online. He was on board of. He was the only actually Israeli on board of an IODP cruise, research cruise on the Maldives, without being in the Maldives physically, but he was on the boat for 2 months, and maybe he will explain about the IODP. It's a very big international community of ocean research that Israel was a member for, and he was the first and the only so far Israeli on such an experience, and involved in many associations and activities related to sedimentology, carbonate sedimentology, geology in general, and marine geology in special. Okay, so in order to shorten the introduction, I leave the podium to Or, who is going to talk about rethinking the fundamentals of carbonate sedimentology. So thank you very much.
Okay. And good noon, everybody. Hope you managed to get your lunch before the talk. I'm kind of disappointed that we have to do this online. I was kind of expecting to come up to campus, see everybody, but c'est la vie. This is gonna be a bit of a weird talk. I'm gonna start with some concepts and then we're gonna build out from there. So, carbonate sedimentology is a sub-branch within sedimentology that deals specifically with carbonate rocks or carbonate sediments. And we can think of the entire field as sort of a big show. And that show has one leading prima ballerina, and that's reefs. But the show itself is quite big, quite extensive, and there is a lot of other dancers on the stage. And they're not getting as much attention. All of these different things form boxes, which we usually refer to as carbonate faces or facies in short. And that's kind of a problematic and not necessarily straightforward term. And this is kind of where the problem starts. So what exactly is a facies? The original definition is incredibly old. It was set out by Gressly working in Switzerland on the Jurassic rocks back in the 19th century. And he basically defined it as a body of rock with specific characteristics. A few years later, Walter tried to refine that specifically for sediments and defined a sedimentary facies as a distinctive rock unit that formed under certain conditions of sedimentation. You can already start to see the problem. About 100 years later, Eric Flügel was trying to refine this further, specifically for carbonates. He refers to this as microfacies, because for the way he was approaching this, the work was done primarily on fin sections. And his definitions was that this is the total of all sedimentological and paleontological data which can be described and classified from the various methods to look at rock. But facies isn't limited just to hand samples. It's not even limited just to carbonates. We do have the facies we describe in fin sections and peels and slabs, but then we have much, much wider scale. We use the term for seismic analysis. We use the term to define things that are kind of similar, like seafloor types. And all of these things we refer to as facies, which, kind of gets us to a problem with the term because it is very wide, it's poorly defined, and yet it is fundamental for how we're approaching any analysis in the field. And this also links to one of the problems. If we are saying that this term describes things that are informative about the positional environment, we also have to acknowledge that the ability to inform environment is limited by the information we can extract from those samples. If we go back to our show, we are limited in how we can extract information from different things, and some environments have a bias towards giving us more information. And there are other reasons why reef facies receive so much attention. I mean, other than the fact that modern reefs make for a great diving destination, but let's not assume that everybody who studied reef doing it only because they want to get paid a holiday on research budget. Reefs, at the end of the day, are really important for biodiversity. This is a fairly recent paper by Closet, and what they're showing here is that if we look at reefs, the richness in reef is so much higher than any other type of deposit, and this has been consistent for the last 500 something million years. So for biodiversity, they are very important. But generally, right now, tropical shelves are where most carbonate are accumulate. Almost half of modern carbonates accumulate on tropical shelves. This is from a compilation we're currently preparing to publish. But even on tropical shelf, let's go to the Great Barrier Reef, possibly the biggest reef system on Earth. That is only 9% of the northeast continental shelf of Australia. So if reefs are something we know a lot about, but are only a small part of the production of carbonates, what's everything else? Answering this question is a lot easier in the past, actually, than in the present. And one of the reasons for that is because working at sea is expensive. Working at sea is hard. It's a lot easier to go to outcrops and collect information about them, assuming they exist. Giovanni Coletti, who's been my longtime collaborator on many of this stuff, and I, at a certain point, decided let's try that. Let's look at what most of the carbonates are made of. We went on and looked at the Cenozoic of the Middle East. Why? Because it's relatively well described, well accessible. There's a lot of studies about it, and they cover a wide range of periods. We were able to map out quite a great number of outcrops throughout the Middle East, going all the way to India. And what we did for each of these outcrops was we went to the papers and painstakingly went over each and every paper to find what were the faces they described, what were the components and the features in those outcrops. And for each faces, we mapped how much of the volume of the unit is captured by that faces. And we went and we looked at their description and we looked at their photos and we reclassified all these different faces into new groups based on what were the principal elements in each one, and then ascribe to that the volume for the unit. So for this outcrop, for example, most of the outcrop is made of faces which are dominated by large benthic foraminifera, or LBF here. And then a little bit have faces which also have red calcareous algae, or a combination of red calcareous algae and large benthic foraminifera, and a tiny bit of them are faces dominated by small benthic foraminifera. And this was actually quite representative of most of the outcrops we analyzed. So this box plot describes, gives you the range of how much of the volume of the units for each time period is dominated by these large benthic foraminifera dominated faces, or co-dominated, so large benthic foraminifera and something else. And the range is quite big, but we can see that throughout the Cenozoic, large benthic foraminifera dominated faces were very common. In some cases, they make up for the bulk majority of the units. In the Eocene, on average, they make over 85% of the rock units, and over half in the Oligocene, and about 30%, and just over 30% in the Miocene, a little bit less in the Paleocene, but we don't have as many outcrops in the Paleocene, so that's not as well represented. This was actually quite surprising because, again, everybody keeps talking to us about reefs, and reefs are described quite differently. So let's take the Eocene, the period where, when we look at volume, most of the volume is dominated by large benthic foraminifera. So what we have on the left is the volume estimate for each one of these dominant faces component from our compilation for the period between 55 and 35. Why specifically that? That's because that's the period where we had the most outcrops, so we could do the best statistics. On the left is a database called PaleoReef. It's an amazing database which compiles the dominant reef builders for each reef that has been described and cataloged. It's currently operated by Wolfgang Kiesling at the University of Erlangen, who's doing really, really important work there. And what we can see is quite stark. If you look at the left plot, then you can see that the dominant component in most of the reefs are corals. Foraminifera are present, but they're not dominant. But if we look at the rock volume, foraminifera are extremely dominant. We have a discrepancy. If we look specifically at the corals, when we looked at the rock volume through the entire Cenozoic, on average, corals make up for less than 5% of the rock volume. But they make up for more than 60% of the reefs. Another thing that is quite common in these reef databases are red coralline algae or red calcareous algae, which were barely present as the dominant component of the rock. They were present in significant quantities in the Paleocene, Oligocene, and also the Miocene, for example, as something that was co-dominant. So something else was the dominant component, and also they were present. And they're quite important because these are binders, and they keep the sediment in place so the potential for preservation increases. But they're not making a lot of noise themselves. So from our analysis, we do get something very clear. Carbonate production, even in warm, shallow seas, is not just reef. And we do see an outsized role of foraminifera. But knowing what the dominant element in the phase seas are doesn't mean we have a good control on the environmental implication. Remind you that the definitions that Walter was presenting were saying that these were things that were informing us about common depositional settings. Going back to the paper I showed earlier when I talked about different types of phase seas, this is a study I did in Malta a couple of years ago, where we had excellent habitat mapping. We had really, really detailed multi-beam and backscatter data for the entire shelf. We had a bit of seismics. And we knew exactly what ecotopes we're sampling. We had wave data, so I could delineate which were wave-dominated environments. And not only that, I could more or less tell you which one were in the above the fairweather wave basin and above the storm wave basin. And about 90% was the cutoff I used. I actually probably should go back and talk about this for a little bit. The storm wave base and the fairweather wave base are these terms that are being thrown around in sedimentology in general, especially when you look at outcrops to say, oh, this is shallow water, it's above the fairweather base. Oh, this is mesophotic because it's between the fairweather base and the storm wave base. This is deep water because it's below the storm wave base. Okay, here's the thing. These terms are arbitrary and meaningless. This is something Peters and Luce already said, by the way, about a decade ago. They analyzed wave data from the east coast of the US and the Gulf of insert your name here. And they showed that there is no clear delineation between storm wave base and surface waves. This is before we start talking about internal waves, and we'll get to that later. There is a continuum. If we still want to use the term fairweather wave base, we need to make some sort of delineation, an artificial delineation. I suggest the 90th percentile, which for the Mediterranean is about 25 meters, for the Caribbean is about 50 meters, and for the East Atlantic, where they have all the hurricanes, that's about 100 meters. It's something, I still want to develop this further, but that's a down the line thing. As a side note, and I'll probably get back to that later, I'll remark that just because surface waves aren't reaching the seafloor doesn't mean that there isn't a seafloor to be shaped, and or to be precise, doesn't mean there aren't waves shaping the seafloor. They just aren't necessarily surface waves. These are not the waves we measure by satellites and buoy, but things that are sitting further down, like internal waves. And we see this here off Bustana Galil, where we have high-resolution seafloor images. We do see bedform formation, which are coarse-grained and cannot be explained by surface waves. That is something that requires a bit of reevaluation in paleo, because there's a lot of arm wave about internal waves there. We don't necessarily know how to interpret this because, again, we have problems with how we interpret faces. Going back to Malta, when we looked at the habitat mapping, there were three main domains: seagrass meadows, sand flats, and marl beds. And we looked, and I ran transects through all these different faces, sampled everything, we made thin sections, we looked at them under the microscopes. And we did something that's a bit different than the way people usually do this. So people usually give you a qualitative description. We found a calcareous sandstone with some coralline algae and some foraminifera. That's what we usually found in the description when you look at the paper. We went one step further and did this quantitatively. So sat down, point counted all these thin sections, quantified everything, and when you have things that are quantified and environments which you have good control on because it's the modern and we have monitoring, we can start doing some statistics. Well, we can try and do statistics. The results were annoying. Working with fully quantitative data, we can see that there is some separation. The sand flats, the seagrass meadows, and the marl samples do separate on multidimensional spaces, which is what you're seeing here. This is a DCA and an NMDS analysis for the data. The axes are kind of arbitrary. They're a stack of the various vectors. But you can think of this as a 2D projection of an n-dimensional map where the different things that build together that are getting plotted on top of each other. The axis 1 is the combined vector set with the most variance, and axis 2 is the second most high variance, and so on and so forth, and there can be a lot of axes. And in these maps, we can see that the seagrass meadows and the marls do separate, but the sand flats are kind of intermix in between. And it doesn't even matter if we are looking at something that's sandy or gravelly, we're getting different quantitative faces as it retains to what makes them, what are the components. The marl, of course, have a lot of RCA, red calcareous algae, but everything else is scattered all over the place. And that's just one location. Surely, if we look at the things which make up the faces across the entire planet, we could find some generalized rules we could use to constrain and better calibrate faces model. So when we try to do this in the past, we have anchors in the present. Of course, we do have interpretations for ancient faces. The problem is that a lot of these were delineated 50 years ago, 100 years ago, based on very anecdotal data, and sometimes kind of like a gut feeling, it feels like. So let's try to do this a priori. This map delineates the light zones on the ocean floor. The pale blue line delineates the 0.05% of photosynthetic available radiation at the seafloor. That means below, at depths greater than that line, there's no light at all. The yellow line delineates the base of the euphotic zone, according to some definition. In between with them, we have the mesophotic zone. These are the light zones, and if we say, well, we don't want to get too complicated with all the deep water planktonic versus neritic factories. Let's just look at where there's light, because where there's light, there is prime productivity. We went out and sampled a few locations, but that wasn't enough. So we went to the literature. And we spent, I don't want to even talk about how many hours because this was a deep, deep rabbit hole. I found myself dragging multiple people down. We began to collect and digitize every bit of shallow water assemblage and facies data on the modern seafloor we could get our hands on. Some were easier. Ebert Gischler specifically does amazing work in the low latitudes, is very good about publishing his data. I wish more people would take the way he works as a model. Others, I won't name, were not so easy. We spend a lot of time digitizing graphs and tables, some old, some very recent. By 2023, we had about 3,000 data points. Not all of them fully quantitatives. Only the green points on the map we had quantitative data for. The red ones we had information for, we had description for, but no quantitative data. And I'll get back to some of that later. I would note that we haven't stopped working. Giovanni and I have a PhD student now, Giulia Galimberti, who's still working on adding more information to this database and doing statistical analysis on that. So if before that we had 3,000 data points, and out of which about 1,500 were fully quantitative, now we have 4,000 quantitative data points, and there is more coming from new locations we've sampled and analyzed. What we're doing now is we're moving another step forward, and we're looking also at about geochemical parameters and hydrodynamic parameters, which we couldn't do before. But that's work in progress. In a few years, maybe we can get Julia here to talk about those. But we had a database, and that's already great because we now have quantitative data. And quantitative data, we can start looking at analysis. We can start looking at relationship. Look at distribution at space and relate one to the other. Just for demonstrative purposes, I plotted up the coralline and the corals and coralline algae data from all of these up here. And they correlate to some extent. They do have this nice line above which both of them aren't present. Below that, it gets a bit complicated. So now we have all this information, and we want to be able to go from the components, from the observation, to environmental interpretation, or at least relate the assemblage to the environment. So we need environmental data, and we need consistent environmental data that we have long time series for. We see an average across all geographic locations, which is a problem because in a lot of these locations, somebody went, they did a cruise at a certain season, they have some information, but for the rest of the years, we don't have information. When we did this paper in 2023, I didn't want to go use models and reanalysis. I wanted to use empirical data. And we needed solid empirical data with long time series, and the only way we could do that was with satellites. And for the record, all of this data and all these scripts are available with redundancy online, including the computation of the workflow and other analysis we did. So if you want to look at the data, look at the inputs, look at the scripts, they're out there, and you can reach out for them. So now we can do something really cool. We can be a priori. We can ignore all previous assumption, all previous knowledge, and just naively take the data. We have the physical description of the samples, the environmental information we got from the satellites, and start splicing the facies based on the abiotic parameters. And we can see, for example, that if we take for one set of parameter, which is which climate zone belt we're in, As we all were taught in undergraduate, there's a shift in composition from the tropics to the high latitudes, going from corals being very important contributors in the tropics to being completely absent in the higher latitude. Filter feeders are much more abundant in the higher latitudes, while coralline algae and mollusks are basically everywhere. And that's already where things start to break apart. Looking at this information at the single variable level is not granular enough. We can't do inversions based on linear relationship. We need proper multivariate analysis, because if we look at the data and we try and look, and here I'm showing a correlation matrix of all the environmental variables and the various components we found that we had descriptions for, you can see most of these relationships have very low correlation coefficients. And even the ones that have like relatively good correlation coefficients, for example, SBCC stands for colonial corals, they relate to surface temperatures and water depth and energy availability at depth. In theory, when we look at these simple numbers. But then when we plot them up, yeah, those relationships don't really work. So we have to move to again back to multivariate space. Let me walk you through what's going on here, because it's a lot. In the top, we have the data points. Color are just for visualization. Brighter are shallower, darker are deeper. And we can already see things are kind of messy here. And the different shapes delineate which climate belt we are. The dots are tropical, the diamonds are subtropical, the squares are temperate, and the triangles are cold, which we didn't have that many back in the day. The lines are the vectors for the abiotic parameters. These are the variables we got, like temperature and radiation and other depth, latitude, radiation at seafloor, chlorophyll abundance. And they're pulling the data in different directions. Latitude and chlorophyll and turbidity and water depth all pull on one direction. And then sea surface temperature and SW, which stands for the amount of radiation available for photosynthesis at the seafloor, pull in the other. And we don't really see any differentiation. We don't see the different points forming clusters. They form a continuum. We don't see any separation. There are these two terms in carbon sedimentology, the T factory and the C factory. The T factory stands for the tropics. These are the coral reef builders and the Uai chulls and all of that sort of stuff. And then the C type factory is for the higher latitudes, cooler, temperate factories. These are the coralline algae and bryozoan and all of that sort of stuff. And they're not separated. They continue from one to the other. If we look at the distribution, we look at the tropics called stuff. This is our T factory, that's on one end, and our C factory, well, that's on the other side. But they never separate. And this is kind of one of the important points I want to pull out of this. A carbonate facies is not something discrete. It's a segment along an ecological gradient. Using a different type of analysis and projection, I think we see this much clearer, where we have this wide ecospace going from warm and well illuminated to dark and cold, And it doesn't matter if we generate this gradient by going from shallow water to deep water or going from low latitude to high latitudes, they converge. There is a non-uniqueness. This means in theory, by the way, that if we want to say that we go from a coral reef faces at the bottom of our section, and we go up into a coralline dominated faces and eventually to serpulate faces, which is kind of a shift from a C-type factory to a T-type factory. Conventional wisdom back in traditional interpretation would be that this represents a sea level rise. But it can also represent cooling. It can also represent eutrophication. What this means is that we need to start thinking more like ecologists when we want to interpret ancient carbonates. Less trying to fit things into boxes and more looking at gradients. Because carbonate faces is an expression of energy availability. Energy is key here. We have non-uniqueness, but if we look at this from a perspective of energy, how much photic energy we have in the highlights, how much photic energy we have at depth, how much metabolic available energy we have at shallow tropical environment, it starts to make sense on why we're getting this continuum. Because energy is the key currency in the natural world. And I can already hear some complaint on this because I'm talking about things in the modern. And the modern is a bit different from the past. Not because the world is different, but because the way things get preserved. Most things that are living on the modern seafloor will not get preserved, especially if they're made of aragonite, which is one of the most common carbonate minerals being produced in the ocean now. So the living assemblage, if we look at it, oh, it's very wide, it's very diverse. But the diagenetic assemblage, the post-burial assemblage, that one's a lot scarcer and sparser. So let's look at what's going on in the Pelican. We wanted to test this. And we needed a case study. Since I've been working with Italians and Spaniards on this, of course, we were drawn to look at the Mediterranean. Because what we want is something with a lot of data, well-established strong abiotic gradients, where we don't need to guesstimate too much about what was going on with that. Lots of carbonates. Preferably it won't be too old, so we don't need to deal with things like evolution and changes with ocean chemistry. And as it happens, the late Miocene of the Mediterranean fits all these boxes. Well, almost all of them. There is one thing that we also need. We need good descriptive data, preferably quantitative data. And there are a lot of papers describing these late Miocene outcrops in the Mediterranean, but they're not that detailed. We looked at all the reefs described in the late Miocene of the Mediterranean, and for most of them, the best we could do is say, was this group reported or not? Present absence. And what we got was a mess. There are gradients in there. And we do see a differentiation in the vectors of the components, but it's going all over the place. We can't really differentiate the sections. Also, we can't really differentiate the different parts of the Mediterranean where we know there's an ecological gradient. And this is without even touching something we realized as we later on, which is there are things that are just underreported, like crabs. And we don't know if they're underreported because they weren't there or people just didn't report them. So that left us only one option. We got samples, made spin section, got on a microscope, and generated our own quantitative data. We are able to get and analyze so far 5 sections, 2 from the Eastern Mediterranean, from Cyprus and Eratosthenes, 2 from the Central Mediterranean, Aquabona, which was one of the sections I showed earlier, and another one from Salento, and one from the Western Mediterranean, from Sorbos. And by the way, I'm showing all this multivariate analysis with different tools and different approach. There is a paper Emilia Orchowska and I wrote a couple of years ago explaining how to apply these types of statistical tools to sedimentological work. And in the supplement, you can also find the R codes if you want to do this analysis yourself. Back to the data. I'm going to focus on the non-metric dimensional scaling and the NMDS because I think it shows differentiation the best. And while the differentiation is not clear, we can see that the Sorbos sample cluster more in the lower left, and the Eratosthenes sample cluster in the upper right, which is good because one of them is on one side of the grain and the other one is not the other side of the grain. So we kind of want them to separate. One of the interesting things for me was that we didn't see a clear division of phototrophs and mixotrophs. Axis 1 is the one with the most variance, and we can see that along it, these groups split. On one side, we have the green algae and the large benthic foraminifera. And on the other side, we have the colonial corals and encrusting foraminifera, and also the red calcareous algae, but they're kind of all over the place, so they're not really distinctive. But this is not a gradient where we see the split. That split falls on a different axis. We seem to have a combination here of axes along which there is a differentiation because we do have two superimposed gradients. One is a spatial one, and one is a temporal one. We do said that the samples from the West Med are specifically ones which have sample species that relate to more biochemical and energy sources, while the Eastern Mediterranean has more photonic energy dominated species. So more heterotrophic versus more oligotrophic. Kind of the opposite of what you would expect, but this is what we're seeing. And there is an internal gradient between these two end members. But there's also one other element forcing this. And that's time. If I take the samples we analyzed, here I'm showing the samples from Aquabona and the Eratosthenes seamount, and I split them into 3 intervals along the section, from the base, the middle, and the top of the section. One of the things we can see is that the relationship changes with time. And that adds a wrinkle to this analysis, because we're not looking at time equivalent spaces. We're looking at a combined effect of changes of environment with time, with an ongoing strong ecological gradient due to the basin being restricted. So when we interpret the dataset like this, there are several dimensions that change the record by phases. And that's something we need to keep in mind and need to further develop the tools to fully delineate. Because at the end of the day, all models are wrong, some are useful. So, take-home message from all of this. The interpretation of carbonate facies and microfacies has to be done careful and conscientious of this non-uniqueness. Quantitative work helps with further differentiation and inference of gradients, but the carbonate facies themselves are segments along this ecological abiotic gradients. They're not discrete entities, which make their delineation either arbitrary or fuzzy. The underlying governing factors on these facies, the factors generating these gradients seem to be dominantly metabolic energy as a key regulator. And I'll call that a wrap. There are a lot of people contributing to this. I mentioned some of them through the talk, and I tried to actually organize all of it, and I realized I'm going to forget somebody. So everybody who contributed this over the years, thank you, are thanked. And with that, I think we'll open the stage for questions.
Thank you very much, all. Congrats for the very thoughtful talk on this. And I open the podium for questions from the audience. Come on, don't be shy.
Hi. Hi, Or. Hi, Nicholas. I don't have a lot of questions, but some clarification maybe. So in general, I have a sedimentological background, and our lecturers from my bachelor and master, every time they said that facies, it is body rock, which is has fingerprints, or how to say it, not describe, how to say, fingerprints of environment. So in general, if you say, I don't know, because facies and this topic is really like philosophy maybe, and a lot of scientists have their own opinion about this, but in general, I think that facies, it is rock, which should have like factors, or how to say, fingerprints of environment. So when you call facies, you need to say and rock and environment. So I just wanted to say, ask, what do you think about this? I mean, facies, for example, facies of quartz, sand of the beach, or something like this. So, and once I failed my exam just because of in lithology and sedimentology, when our lecturers asked us, call these facies, and I didn't said that this is sand of the beach. So, and that's why when you talked about even microfacies, you talked and mentioned foraminifera or coralline, but it is in general is just a component classed or something like a rock. So what do you think about this facies concept again? Because I didn't clear, so you understand, I didn't...
Yeah, I get what you're saying. Well, my approach is that using the term facies to say this rock is this environment is kind of an archaic approach. This is something that was delineated in 1893 as a concept and did not get a lot of revisitation or reevaluation since. And people have kind of used this as gospel, where in practice, what we're seeing when we look here and we try to quantify this, and this has been done, by the way, long time ago for siliciclastic facies, when you try to take a body of rock and say, this thing is this environment, when you look in the outcrop, nobody can tell you you're wrong. But if you go and you go, okay, what is this thing? I have a quartz sand here with hummocky crossbedding, and it has a grain size of about 0.3 millimeters to 0.5 millimeters. And you go and you look at all of the possible environments that have those combinations in the ocean, you're going to find that there are multiple environments that have that combination. So if I look at the outcrop, I can tell you that it's definitely a quartz sand with a chromic crossbedding with this grain size. But telling you what the environment is would be folly, because non-uniqueness is an inherent feature of sedimentary deposits. We can get the same output from different set of parameters. And the result of that is that if I'm looking at the outcrop, I can tell you what I see, but the interpretation is subjective and it can be biased by, this is what my professor told me, and this is what his professor told him about this unit, not necessarily by what is actually going on there and how it fits into the entire succession. My approach to faces a description, and this has been my approach since my PhD, is that we first delineate faces based on things we can describe and quantify. This is not something I came up with. This has been the dominant approach in Europe for at least the last 30 years. And then once we have the description, we try to ascribe an interpretation based on what we have in each group and how it relates to all the other groups around it in a way that makes sense. Because if, for example, we have I'll try to keep it simple. If we have a succession with paleosols, which are quite strictly identifiable, and in between we have these cross-bedded sandstones, I think it would make sense we can interpret these as either eolianites, so like dunes, or coastal deposits, because we're still more or less in the same set of environments. And then we can try and break it down based on dune structures and see if this makes sense to be something that is wind deposited or wave deposited. But then you need to look at this from a broader sense. You can't just look at the one rock and interpret it. It has to have context, and it has to have a broader, and it has to be done with some thought. So I think the two things kind of need to be separated between let's say faces description and faces analysis or faces interpretation.
Yes, I understood what you're talking about, but in general, of course, we can't look at the rock separately between take out from the system. We need to see like a broad system, and if you have a rock, you need to see what is nearby of this rock and etc. But also about quantification. So in general, you said for me it's surprised that the quantification is not how to say it, popular because in 2018 I worked with carbonates, and we used quantification. We described carbonates just in thin section. I've never saw this core, and I had just thin sections, and we described this thin section by primary features and secondary features, and you need to describe it by percent. And my question is, what is the approach for quantification? How you can quantify except this percent?
Well, you can do the percents in different ways, and I'm not sure. I don't have the figures here, but we had a master's student at some point looking at the difference between percent volume, which was very annoying to differentiate, because basically what we did is we scanned the entire thin sections, and then they basically sat in GIS and made polygons of everything to measure the percent volume. And then we also did point counting to compare it to that. And there are, by the way, I know of at least 3 different approaches to point counting. All of them result in something different. But the end result is actually that we found that the differences are actually quite small. There is a difference between percent mass, but percent mass is something you can't really do in paleo. You can do it with the modern samples because we can separate them and we can weigh them. But once it's solidified, separating them for weight is not possible. But there are still a lot of papers being published now. I'm telling you because I'm like Nico mentioned earlier, I'm an editor, and I also review a lot of papers, so I get to see some stuff that's being submitted. And there is still quite a bit of papers which are fully qualitative. It's not it's an approach I don't say, I won't say it's necessarily something we need to get rid of. There is a point to this qualitative interpretation, especially when there's a large sample set that needs to be described and the time is short. So it has its place, but it's still quite common.
Yes, it's really interesting because approach should be like common for all of us, I think, for sedimentologists, I mean. So thank you, Or. It's a really nice and interesting presentation.
Okay, thank you, Alexandra. Are there other people that they want to ask questions or comments? Well, so if everything is clear, so I would like to thank you, everybody. And thank you again, Or. And looking forward for the next meeting. Okay.
Thank you. Bye.
Bye, everybody.
Thank you.
Bye.
Bye-bye.
Bye, thank you.
Thank you.
Itzik, are you here?
כן, אני עוד פה.
אוקיי, אני אתפוס לך אחר כך.
אני הולך להביא את האוטו שלי, ואז אולי אני אקפוץ אליך. אני אדבר איתך כשאני מביא את האוטו.
אין בעיה, יאללה ביי.
ביי.